Good afternoon, and thank you for joining us for Meet the Minds Behind the Mission, a live Q&A with ADDF scientists. For those of you who I do not know, my name is Katie Pizzone. I'm the Senior Director of Philanthropy at the ADDF, and I'm delighted to be joined by two ADDF scientists who are leaders at the ADDF and will be sharing their perspective with you today.
Thanks, Katie. Great to be here today. I'm Aaron Burstein, Head of Search and Evaluation for the therapeutics team.
And I'm Melissa Lee. I'm Director of the Diagnostics Accelerator, which you'll also hear us refer to as the DXA.
Thank you both. Before we kick off our conversation, one quick housekeeping note. This is a live Q&A. If you do have questions, please put them in the chat box at the bottom of your screen, and we will have time to get to those in a few minutes, actually. So in honor of World Alzheimer's Day, we wanted to organize this conversation to highlight the most recent advances in Alzheimer's research and what is most promising in the field to come. As many of you know, the ADDF's model is unique and places us in an ecosystem bringing together venture capital, the world's leading philanthropists, and the brightest minds in academic scientists. We take risks when investing in the most innovative science that industry or other funders shy away from. Our team brings industry rigor to scientific diligence, and we make investments, not grants, making sure that the best science is funded and any returns go right back into our scientific pipeline. Okay, so I'm going to start with a question for both of you today, and these are some questions we received in advance. This one's for both of you. I know that working closely with you both, it's a great privilege for me to spotlight the work of you and your teams. Pulling back the curtain, what does a regular day look like at the ADDF as you find and fund the best science? Aaron, I'll start with you.
Thanks, Katie. So I lead a team of scientists who are focused on what we call our therapeutics portfolio, which includes our funding programs to pursue drug development, as well as biomarker development in cerebrospinal fluid and neuroimaging tests. Our group is focused on drug development initiatives, as well as building strategic partnerships to help advance the development of new therapeutics for Alzheimer's disease. Much of our day is spent pursuing opportunities, whether it be through outreach to academic or biotech investigators based upon what we read in the literature or what we see in press releases, or inbound interest from those individuals looking for potential funding for their projects. We actually support a number of programs through our drug development CSF neuroimaging calls for proposals, but also beyond Alzheimer's disease, we support drug development for other related dementias. We have one partnership with our colleagues at the Association for Frontotemporal Degeneration, which is a funding program to advance therapeutics specifically for FTD disorders. We have another funding program with the Harrington Discovery Institute focused on identifying innovative academic work, and through funding through ADDF and scientific and technical support through the Harrington Institute, we can advance the therapeutics that are being developed by academicians. On any given day, we're likely reviewing a proposal. In a year, we see upwards of 300 applications for our consideration. So a lot of our time is spent reviewing the science and making determinations around where to deploy funding to advance research. Melissa, would you like to speak about your day?
Yeah, well, our day-to-day is not all that dissimilar from your team, although the DXA does work a bit differently than as your peer because we operate as a $100 million investment fund that is funded up front. It was started back in 2018 by Leonard Lauder at the ADDF and Bill Gates at Gates Ventures. And this was back when the field wasn't sure if a blood test for Alzheimer's was even possible. And so it is very specifically focused on accelerating the development and commercialization of biomarkers and diagnostics that can be early, accessible, and accurate. So I specifically lead the DXA AI and digital portfolio, And I also lead our flagship digital data collection program at the DXA that's focused on speech. It's called Speech DX. You'll probably hear me talk about it in a little bit. Our teams do intersect quite a bit, though. And so we have our weekly meetings with the full science team across all of our departments to talk through the science we're working through at the moment. And sometimes things will come across my desk that don't fit specifically into the DXA funding priorities, but they might fit a little bit more into Erin's groups. And so we'll pass things back and forth as relevant.
Thank you both. There have been so many exciting milestones in 2025. The first FDA-approved blood test for Alzheimer's disease, also a subcutaneous administration of Leqembi, also AI and digital advancements, which will enable early detection and diagnosis of Alzheimer's disease. Melissa, as the expert in this area, it does feel like AI is everywhere and on everyone's minds. Tell us how AI tools and new technology will lead the way in the future of Alzheimer's diagnosis and prognosis.
Yeah, AI really is everywhere these days, but we've actually been investing in AI for a long time now, pretty much since we started the DXA, because it's always been clear to us at DXA that even though a blood test to tell you whether or not someone has amyloid in their brain is a huge, huge step forward, I can't really overstate how big of a deal that is. It's still not the full story. You can't tell how someone's cognition is functioning via a blood test. And the tools that we have for this today are quite bad. So digital tools and AI are absolutely critical, we think, for solving that. Our biggest investment in the space is in speech, which we believe to be hugely promising for detecting Alzheimer's early and accessibly, and even for predicting future cognitive decline. I can get into that more in the future if we want. But AI is really all about data, and a model can only be as good as the data that goes into it. We think speech models have so much potential, and so we decided a few years ago, actually, to invest in the space by building the largest longitudinal high-quality speech and biomarker data set in the world that other groups, companies can use to develop these speech-based AI models. And so this is the Speech DX program that I mentioned earlier. Been working on this for a few years now, and I'm really excited that as of this year, we are open for business. So we just went live with our first release this summer to our first commercial partners, Siemens Healthineers and Calliope, who are using this data set now to start building these speech AI tools for Alzheimer's. And we have more partnerships coming. And so if you're in the audience and are interested in licensing this data for your company, just email me and let me know.
Thank you, Melissa. So exciting. And we know that the earlier we can diagnose Alzheimer's disease, the better chance that these therapeutics can be used as interventions for patients. Tell me a little bit more about how these new tools will be impactful for patients in a clinical setting.
Yeah, well, we had, as you said, a real breakthrough moment this year, and for the first time ever, the FDA cleared a blood test for Alzheimer's. This was a blood test from Fuji Radio, which we supported at the Diagnostics Accelerator. Again, this is something that scientists weren't even sure was possible back when the DXA was started. There were actually op-eds by very prominent people in the field at that time saying that it wasn't. So this is a huge moment for the field, and it means that these patients can get these tests all over the place now. And I'm actually curious, Erin, how you see this from your point of view.
Yeah, I mean, I think the expanding biomarkers are really exciting, that the scientific innovations lead to the regulatory approvals and ultimately the prescription or third-party payer coverage that allows for the clinical use of these agents and the payment for it. We have been interested in this area for decades, and actually were a seed investor in a company called C2N, who had actually developed the first blood test for Alzheimer's disease. What we're seeing is the market expansion and growth of the research in this field, leading to where we are today, as you've described, with the approval of biomarkers that we can actually now use to look at a blood test. And we're eagerly awaiting decisions from CMS around reimbursement for tests such as the Lumipulse test that you described. This is, in some ways, if you look at other fields, we're at a point where it's analogous to cardiovascular disease was when we realized that we could look at cholesterol in blood and identify patients that had high LDL, and those being patients that could subsequently be put on pharmacologic therapy. We're at that stage now where we can actually identify patients with the underlying pathology of interest and put them on the appropriate treatments for slowing the progression of their disease.
Yeah, and I think that's really where we're at right now in kind of the immediate term, and in the near to medium future. I think blood biomarkers, we also see them working with these digital measures like speech to give you a more complete measure of what an individual's case of Alzheimer's looks like. And then beyond that, in the longer term or slightly longer term, we are investing in a future where blood tests can be paired with digital AI tools as part of routine screening, or in digital tools that can even be used before you go to a doctor's office that are passively detecting your function as you go about your everyday life, and even letting you know if there's a reason for you to go to the doctor.
Yeah, and Melissa, I think as today we're talking about symptomatic patients, but as we think about moving into prevention and the need for biomarkers like you're describing that can identify patients who perhaps have the underlying pathophysiology of interest but aren't symptomatic, there really is a tremendous need for biomarkers in that space, and would be interested in your thoughts on sort of where we are with biomarkers that could be used for identifying patients that could be included in prevention trials.
Yeah, well, I think this is a place where AI is actually really exciting. So one thing that AI is really good at doing is taking really large amounts of data and picking out really small signals from all of the noise in that large amounts of data, and tell us something about something that we as humans looking at it, combing through the data ourselves, wouldn't be able to pick out on our own. And so I think one of the things that is starting to happen, it is just now starting to happen, but it's a little bit further advanced in the cancer space, but now starting to happen in the Alzheimer's space with these AI tools, is the integration of all of the information that we already have on a patient in their electronic health record, into something that can say something about an individual's risk for developing Alzheimer's, so that you might be able to know who is more suited for one prevention trial or another.
That's great. That's so great. Let's go back to the FDA, because I know that people hear FDA and want to know everything. So I know another recent FDA approval was the use of Leqembi in a subcutaneous delivery. Erin, can you tell us a bit more about that? But also, please remind our audience what subcutaneous is.
Sure. So I'll start with what is subcutaneous. And this is basically giving an injection under the skin. So much like when you go in for a vaccine, while slightly different, it's that type of injection where it doesn't require going into a vein, but can just be injected just underneath the skin. And this represents a tremendous opportunity to simplify the drug treatment paradigm with these anti-amyloid therapies. 2 years ago, we saw the major advance with Leqembi being approved by the FDA, first through accelerated approval, then full approval. But with that approval was the ability to get intravenous infusions. So going into doctor's office or an infusion center, having an IV inserted, and then receiving over the course of an hour the injection of the medication. And that needed to be done every 2 weeks for 18 months. The burden of going in every 2 weeks, sitting for an hour, the follow-up and monitoring by nursing staff before one can go home. Perhaps valuable because of the modest benefits you can see, but a burden nevertheless. Subcutaneous administration now allows for patients and caregivers to administer the medication at home. The Leqembi in particular is administered through what's called an auto-injector, a device that you basically just press against the skin, and it automatically injects the drug over 10 to 15 seconds. And this can be done at home, much like we've been doing with the GLP-1 receptor agonists or GLP-1s for weight loss. So the Ozempics that we're all familiar with because we see the commercials all over our television, we've been doing this for weight loss for years. And now we have the opportunity in patients with Alzheimer's disease to, in the comfort of one's home, inject these medications. Now, this is approved as what's called a maintenance therapy. So for the first 18 months, you receive the IV administration every 2 weeks. Prior to the subcutaneous administration, at the end of 18 months, you would still need to go for IV infusions, but on a frequency of once a month. While a lower burden, you still need to go to an infusion center and the time involved. This now allows you, after 18 months, to simply administer the injection at home. And I think what's really exciting about this is not only the immediate benefits on patients and caregivers in terms of minimizing the burden of drug treatment, but the next logical step would be a point where you start somebody on Leqembi and it's with subcutaneous administration right from the outset. And if we look at the path with the GLP-1s, aspirationally, one could envision a day when these injectable therapies could one day turn into oral therapies for targeting amyloid. And so that, I think, is, the future that is out in front of us.
Yeah, I'm actually really curious, Aaron, how you see this as something that affects the possibility of combination therapy, if this makes it more or less realistic going forward.
Yeah, thanks, Melissa. So I think it absolutely helps to facilitate combination therapy with anti-amyloid therapies. Many of the projects that we see here at ADDF and are currently in the Alzheimer's disease pipeline are also injections. So IV infusions or perhaps injectable gene therapies. And so combination therapy under those scenarios has now doubled the complexity, double the burden. You're now talking about, instead of one infusion, 2 infusions. If you think about a future of combination therapy, much like we've been administering new investigational therapies to patients who are on a background of symptomatic therapies with cholinesterase inhibitors, one could envision a future state where we start exploring combination therapies of novel medications to patients who are already receiving anti-amyloid therapy. The fact that they will be receiving subcutaneous administration allows for simplification of the treatment paradigms if perhaps you need to administer an IV agent along with these agents.
That's great. And I want to go back a little bit to the GLP-1s. They're a very hot topic for everyone, I'm sure, that's in the audience for one reason or another. And the truth is, at the ADDF, it's something we've been following for many, many years and we've been involved with for many years. So Aaron, on the therapeutic side, can you give us a little bit of color on what our role has been in bringing forth the GLP-1s and for Alzheimer's disease and the news that we might be anticipating later this year?
Yeah, happy to. I mean, I think as an organization, we're really excited about the GLP-1s as a target. And historically, we've been particularly interested in funding programs called repurposed drug development programs, which basically means taking a medication that perhaps is approved for a different indication and exploring its use for the treatment or prevention of the progression of Alzheimer's disease. We previously funded an investigator named Paul Edison at Imperial College years ago to study one of the first generation GLP-1s, a drug called liraglutide. And we partially funded him to run a phase 2 study to generate data regarding the potential utility of this class of drugs in treating Alzheimer's disease. again, small study, but the results of which were particularly encouraging around the potential for this class of drugs. What we are about to see in the coming months, perhaps by the end of this year, is the readout of a large phase 3 study being conducted by Novo, exploring the efficacy of Ozempic in patients with Alzheimer's disease, and in particular, an oral route of administration of that agent. We all know how transformative these agents have been for weight loss and how they've been able to help patients with weight loss and, by extension, other aspects of health. And we're now on the cusp of seeing the potential utility of these agents in an adequate and well-controlled trial that will answer the question around, will these, in fact, improve function and cognition in patients as compared to placebo administration. So again, we expect to see some results by the end of the year. So again, a very exciting time for us to see how our earlier work has catalyzed this phase 3 study that is now being conducted, the results of which will be, I think, very informative to the field.
Thank you, Aaron. Very exciting. And looking beyond GLP-1s, I know that the drug development landscape has never been more diverse and has never been more vibrant, with so many drugs in development with multiple targets. What are you most excited about with this next generation of therapeutics?
Yeah, so I think the breadth of targets that are now in the clinic undergoing clinical studies. ADDF's friend and colleague, Dr. Jeff Cummings, as well as our Goods Prize winner, every year publishes his Alzheimer's drug development pipeline report, in which he catalogs what are the projects and drugs that are in clinical studies, ranging from the first administration of humans to these large phase 3 pivotal registration trials. And I think what's particularly exciting is this year's report, which came out in June, highlights that compared to last year, there are 56 new drug trials, 10 of which are now in phase 3, 21 of which are in phase 1. So of those 21, most of those are going to be new drugs, new targets being explored for the first time. What we're also seeing is that 70% of the new drugs in development are targeting novel mechanisms beyond what has been historically an approach targeting amyloid and tau. And this reflects a fundamental transformation in how we target approaching the underlying biological processes that may put one at risk for developing Alzheimer's disease, and really sort of supports the central role that we've had in advancing what many of you have heard over the years as a biology of aging approach, recognizing that aging is the number one risk factor and the pathophysiology associated with aging can lead to Alzheimer's disease and represent novel drug targets. I think also exciting for us is that combination therapies and trials exploring that are also on the rise, with 11% of all active trials exploring combination therapy. Many of these are exploring two agents combined to see if they can have an additive or synergistic effect on the disease process itself. But there are also a number of combination strategies seeing if they can increase the amount of an active drug that actually gets into the central nervous system or the brain, which is the site of action. There are some medications that are limited by effects or adverse effects that occur, and there are combination therapy strategies looking at ways to reduce the risk of these adverse effects, thereby allowing for dosing higher amounts of the active medication. So again, a very exciting time for us in terms of seeing expansion in the number of novel drug targets, and also recognition that the future for seeing greater efficacy of these treatments reflects the need to pursue combination therapy strategies.
Yeah, and even though the focus of this report really isn't on biomarkers, I also thought it was really exciting to see that it did show that the role of blood biomarkers have really increased in the past year relative to the year prior. And it's, I think, really gratifying for us at ADDF and DXA, and also just very exciting to see that there's now less of a need for these really costly and invasive tools like PET, SANS, or CSF, that were both kind of holding back the application of these potential treatments, but also the trials for them. Though they still have their place, they are now used where they need to be used rather than just everywhere. Because we see these blood-based tests as becoming the standard for a diagnosis in the future, meaning we'll finally have these accessible, affordable, and scalable tools for early detection in standard use. But I think to your point, Aaron, with all these drugs that are now in trials coming online, we need biomarkers to go along with them as well that don't do more than just looking at whether or not someone has amyloid in their brain.
Yeah, thanks, Melissa. I mean, I think there's such an explosion of work in the biomarker space that I think one of the challenges is how does one keep up with all of the ongoing work, the publications, the clinical studies that are now incorporating biomarkers. And here at ADDF, one of the things that we've done is worked with Jeff Cummings to leverage his experience with the clinical trials report to support his efforts in developing something called the Biomarker Observatory. This Biomarker Observatory is intended to be a organized, comprehensive, curated database that can be a reliable and authoritative source on real time. What are the biomarkers that we have today? What are the biomarkers that are being studied? What are their contexts of use? And really provide a resource for those of us involved in drug development as to what do we have and where the unmet needs are. So again, a very exciting time in terms of helping him to develop this database.
Yeah, I think this conversation is so helpful in really showing how the biomarker discovery and the diagnostic development really fuels the drug development pipeline. It's incredible. I want to remind our audience to put your questions in the chat. We will be getting to your live Q&A in a few moments. But back to Melissa. I know that we are heading into our third iteration of the Diagnostics Accelerator, DXA 3.0. And DXA was really built to fill those unmet needs in the biomarker space. And I love seeing your enthusiasm on when we talk about the blood tests. It really shows the real-time impact of the DXA. So my question is, what can you tell us about what the team will be focused on with DXA 3.0?
Yeah, thanks, Katie. As said, we are currently in the process of building DXA 3.0. So I said DXA was a $100 million fund before. We are hopefully that's going to be bigger going forward. And the goal of DXA 3.0, it's focused on enabling precision medicine with the goal of getting more granular than the biomarkers that we have today. So we know that we want to be able to know more about an individual's personal version of Alzheimer's disease in a way that might make it possible to treat that person more precisely. So, including through the work that we've done, DXA 1 and 2, we finally have a good accessible way to tell whether or not someone has amyloid in their brain. So if someone has cognitive deficits, we can reliably tell if those deficits are due to Alzheimer's disease. But Alzheimer's, we know, is a really complex disease. There are other proteins we know that can be misfolded in the brain of someone who has Alzheimer's. And we want to be able to have tests that could reliably detect those different co-pathologies as well. We also believe, as Aaron said, that Alzheimer's is driven by disruptions to the biology of aging. And just as no two people age in the same way, no two people really have Alzheimer's in the same way either. And so we want to be able to know how an individual's biology of aging pathways are specifically disrupted in that individual. And we need to have tests that are able to detect and measure that. And so we'll be making investments focused on all of this, and also building on our existing AI investments as an enabling technology that can ultimately integrate all the different types of data that we can now gather on a person to then tell us, okay, this is what this individual's version of Alzheimer's is. And then ultimately, when we have those drugs to treat that we can combine to treat a person more precisely, how should this individual be treated?
Thank you both. It's now the time to move to our live Q&A, which is very exciting. And please continue to put your questions into the chat, and we will address them as we go. So the first question, I have been tested and do not have the ApoE4 gene. Can I still develop Alzheimer's disease? And Melissa, I will start with you.
Yeah, so ApoE4 is one of the strongest genetic predictors of Alzheimer's disease, but it's not that strong of a genetic predictor for Alzheimer's disease. So if you have two copies of ApoE4, if you have any amount of ApoE4, we know that your risk for developing Alzheimer's disease is higher, but it doesn't mean you will develop Alzheimer's disease. And the flip of that is also true. Just because you don't have ApoE4 doesn't mean that you'll go on to develop Alzheimer's disease. But we do know that ApoE4 is related to Alzheimer's disease, and that people who have ApoE, they are more likely, the risk for developing Alzheimer's is increased relative to people who don't have ApoE4. And there's also evidence from the drugs and drug trials and other treatment trials so far that people who have ApoE4 might respond better or worse to the different therapies. So, for example, the US Pointer study showed that this high intensity lifestyle interventions actually really benefited participants regardless of their ApoE4 carrier status. So everyone can benefit from the lifestyle interventions that were highlighted there.
And Erin, I know we've been funding a program for many years in our portfolio related to genetic risk factors. Can you share a little bit about Lexeo?
Sure. Yeah. I mean, I think historically one would look at ApoE4 and think, well, this is a non-modifiable risk factor. But we've supported this company Lexeo with a really kind of novel gene therapy approach whereby they give an injection of something called a transgene, which facilitates the individual's production of ApoE2. So the study in particular we supported was taking what one would believe to be the highest risk individuals, those that are what's called ApoE44 carriers or ApoE4 homozygotes. And through administering this transgene, what they were able to show in their early studies was that they could actually stimulate the production of ApoE2 and get increased concentrations of this, which one would hypothesize would result in the development of this ApoE2 protective variant, thereby changing someone from a high risk to having the potential to change them to having a more protective state, thereby potentially lowering their risk.
Thank you, Erin. Now, another one for you, Erin. I've heard a lot about inflammation. What role does it play on the onset and development of Alzheimer's disease?
Yeah, this is an interesting question because the answer is it depends when. So, inflammation can actually be a good thing. It's the body's response to injury, and it's the inflammatory process that helps you to clear the insults that are causing damage. So, in some cases, it could be a positive. And in Alzheimer's disease, there are cells called microglia that can actually have a beneficial effect, particularly with these anti-amyloid therapies, in clearing amyloid and other proteins. It's when inflammation goes awry or unchecked that you see an overabundance of these inflammatory processes. And when that happens, it can actually recruit cells that perhaps aren't already in an inflamed state to now express an inflammatory profile, further propagating the development of the disease. So, we, and I think others are very interested in the potential role of using agents that are targeting inflammation to bring it back into more of a physiologic balance to actually prevent the deleterious effects. And we're actually funding a number of programs. One that always comes to mind because it also represents a combination therapy strategy we're funding, Koya Therapeutics, which is developing a therapeutic that takes the cells that regulate the immune system and actually help the immune system and uses their therapeutic to reestablish an appropriate balance to an inflammatory state that is actually helpful in slowing the progression of the disease.
Thank you. Another question for both of you. Will Alzheimer's disease require a multimodal approach, or will there be a silver bullet cure? I'm going to let Melissa take this one first.
Yeah, we do think that Alzheimer's is going to be a multimodal approach, using a combination of therapies, in a personalized way to treat a person's personal version of Alzheimer's disease. And so Alzheimer's is extremely complex. It's any number of aging pathways can be going on to contribute to the disease. As we said, no two people age the same way. They also don't have Alzheimer's in the same way. And so we think that it's not going to be a silver bullet, one drug that treats everyone. We think that what's going to be more helpful actually is treating everyone with their own combination of therapies. Aaron, what is your perspective here?
Yeah, I agree completely. I mean, it is a complex disorder, and I think with the biomarkers that we are learning so much around the variability from patient to patient. When I started doing research in Alzheimer's disease, we had none of these biomarkers. And I know Howard has spoken to this before as well, and that it was based on clinical diagnosis. But what we know now is that some patients may have amyloid and tau, but 40% of Alzheimer's disease patients have what we call mixed pathology. They may have a protein called alpha-synuclein, which we typically think of as related to Parkinson's disease, but there's a percentage of patients that have that protein. Another protein called TDP-43, which we now know a number of patients have. Add to that the underlying biological processes. Some may have amyloid and tau with a high inflammation component. I think the best analogies for this are in the oncology space. There are a number of different tumors one can have, and it's only once you've biopsied the tumor and understand those specific characteristics that you can identify the specific combination of treatments that'll be most effective to that patient. The other example I think of is going back to like the cholesterol and cardiovascular disease. There are a number of risk factors for cardiovascular disease. Some of us have high cholesterol, some of us have high blood pressure, some of us have other risk factors. And whether we're on a blood pressure medication, a cholesterol-lowering medication, both of them is based upon the unique characteristics of each individual. So I think we're on the cusp of really having the tools to move into a stage where precision medicine-based approaches to understanding the specific underlying biology and pathophysiology based on the biomarkers and defining a specific treatment paradigm that's right for that patient's biology is really, I would say, the future, but I think the future isn't very far off.
Yeah,
Yeah,
oh, go ahead. Go ahead, Melissa.
yeah. I was just going to say, I think that's thank you for calling out the role for biomarkers in this area. And I think we actually have an investigator who we funded the DXA who, and so as you said, in the cancer space, you can do things like take a little bit of that tumor, biopsy it. You can't really biopsy the brain in the same way, but we can take things like cerebral spinal fluid, which is still very invasive, still a risky procedure, but you can start to do things like take some cerebral spinal fluid and profile all of the proteins that are within that cerebral spinal fluid. And we have an investigator within our DXA portfolio, Betty Timms, who has been doing that and has found, looking at all of the proteins, so um, thousands of proteins in a number of individual Alzheimer's disease, and then looking how those proteins, those profiles of proteins vary across those individuals. And she's been able to identify, looking at these protein profiles, that there are actually a few groups of individuals with different types of Alzheimer's disease, or what looks like different types of Alzheimer's disease. It's not very practical to have to do this lumbar puncture for everyone. It's not very accessible or scalable. And these really big proteomics assays are that in which you are profiling all of the proteins in someone's body are quite expensive. And so the next step, and what we are hoping to help doing as part of 3.0, is to look at these different protein profiles, and pick out the ones that are really important for contributing to that this kind of flavor of Alzheimer's disease, and then help develop assays that just detect whether or not those really important proteins are there or not, to tell which subtype of Alzheimer's someone might have, in a way that then might tell how that subtype can be treated more specifically.
Yeah, and Melissa, I think bringing it back to something you said at the beginning when we were talking about artificial intelligence, I mean, these large data sets you're talking about, I suspect this is where artificial intelligence could be tremendously
Absolutely.
helpful
Yeah.
in analyzing these.
Yeah.
So I'm going to cut in. This actually is a great segue to another question we received. How does this all work in a primary care setting? If, Melissa, you could look out 5 years from now or 10 years from now, however you'd like to frame it, what biomarkers will we see in our primary care setting when we go to see our doctor? What biomarkers will they be looking at? I know some of this biomarker development is in different stages.
Yeah, well, I think the safest answer I can give you is that in that amount of time, we'll definitely have those Alzheimer's blood tests available within primary care, that tell you whether or not you have Alzheimer's disease if you have cognitive symptoms. The steps beyond that are kind of the tests that can tell you what your cognitive symptoms are. If you have cognitive symptoms, what those cognitive symptoms are. They do go beyond just memory impairment. And then even beyond that, there are tests that can tell you if you might have cognitive impairment, or you might become positive for Alzheimer's disease in the future, that we think are on what we see as on their way now. But I think in terms of blood tests, the next ones are going to be those ones that Erin mentioned, those other co-pathologies that a person can have in their brain. If someone is Alzheimer's positive, there are a number of proteins, alpha-synuclein, TDP-43, that can also be in an Alzheimer's patient brain, that can really contribute to how that disease develops. And I think one of our high priorities at DXA 3.0 is developing assays to detect these other pathologies.
That's great. That's great. And so, Erin, I have one question for you, and then a couple more questions for both of you. So, Erin, what are your thoughts on targeting autophagy for AD treatments? And also, please very quickly tell us what autophagy is.
Sure. So autophagy refers to the sort of natural physiologic process of cell death. So, all of our cells go through a life cycle process where they will naturally die off. And when it becomes problematic is when they don't. And I think Miranda Orr has coined the term, I think, zombie cells, where you get these senescent cells which have not undergone that autophagy or senescence. They go into a cellular senescent state. And the problem is that when you have these cells, normally they would be cleared, but they exist in a state where they may be creating an inflammatory condition, and they're able to recruit other cells to take on that inflammatory state and ultimately lead to the development and progression of Alzheimer's disease. So it's an area that we've been interested in. It's a combination therapy approach that we're funding with Dr. Orr, looking at ways to take these senescent cells and convert them to allow their clearance and prevent the recruitment of other cells to that similar state. We also are very much interested in and actually see a need for additional therapies that target some of the mechanisms behind autophagy. Within the cell, there's a lysosome that's responsible for the breakdown of protein. We don't have right now any therapeutics that target this mechanism for clearing these substrates. And so we're actively looking for programs to potentially support, to potentially augment this mechanism for clearing deleterious proteins and lipids.
Thank you, Aaron. So I'm actually going to skip a little bit to our final question, which is a fun one, and which I think will leave everyone very hopeful about the future. So let's start with Aaron. It's such an exciting time out there in the Alzheimer's field. What are you most excited about? And what will give our audience hope for the future? And then Melissa, will go to you.
Yeah, thanks. So two things actually, and I'd be remiss if I didn't start with the biomarker developments. From a therapeutic side, we see the future as combination therapy, but critical to combination therapy is having the ability to identify the right drug for the right patient at the right stage of their disease. We need biomarkers to be able to do that. And we are seeing that we have some, and there are a number of other exciting developments on the horizon that, again, will allow us to take on a more precision medicine-based approach to how we conduct our clinical trials to answer the question around the potential efficacy of combinations, and then ultimately identify patients who can benefit from approved combination strategies.
Thank you, Aaron. Melissa?
Yeah, thanks, Aaron. I'm also going to cheat and do two things here, I think, since you did. I think I'm obviously hugely encouraged by the explosion in AI that we're seeing in the past months to years. In that these new generative models are, I mean, there's just so much that you can imagine doing with them. But I think one of the things that is really exciting about these generative models is that they are really good at putting together really large amounts of information that and importantly, different kinds of really large amounts of information. And so we already had machine learning AI techniques to analyze large information of one kind. So that proteomics information, for example, or all the proteins, we have techniques for that. These generative models, but it was really hard to take, say, the data from an AI and the data from these proteins and put them together in a way that's meaningful. These newer models are much better at this type of task because they translate everything that they see into one type of language that they then work from. And so I'm really excited in how these newer models can really start to take all of the different type of data and information that we have about a patient and integrate that into something that's actionable for a physician. My second answer is also of course on my pet topic, which is speech. And so one of the things that I think about in the AI space actually is that the models are actually starting to get ahead of where we are with the data, and that as I said, an AI model really can only be as good as the data that goes into it. And these techniques are getting really good, really advanced, but the data that are available for these models to work on are really lagging behind. And so we see speech as extremely promising in this space. It's a huge cognitively complex that we're doing tasks that we're doing all the time. And so it holds a lot of information about your brain and how it's functioning, actually. And there are studies going back to the 1980s that say that there's signal in that speech really early on that tells you whether or not someone might experience cognitive decline. And we have more recent studies saying using retroactive data sets showing that speech can predict future cognitive decline by even up to 16 years. And so we think that there is huge potential. There's a huge amount of scientific potential in speech. And that is kind of paired with the fact that speech is one of the most accessible modalities that you can imagine. We are speaking all the time. I'm literally speaking right now. And not only are we speaking, we're speaking in the presence of microphones that are connected to computational machines at all times. Like I'm speaking to this computer. I have my phone right next to me. I have my smartwatch here. I think probably of those on the screen, we probably have 15 or so of these speech recording computers around us right now. And so it's extremely accessible. It's really easy to capture. And so we are really excited about the possibility of these AI tools being developed and being applied to a data set like SpeechDX that we've been building at ADDF, DXA for a few years now, to actually become something that can detect cognitive decline really early or predict it even before it starts.
Thank you, Melissa, and thank you, Erin. I know that a lot of members of our audience are personally impacted by Alzheimer's disease and related dementias, and I know this conversation has been really hopeful for me and for many of us who have family members impacted. So thank you both, and thank you everyone for joining today. We really couldn't do this critical work without the brilliant minds in our scientific community, as well as our generous supporters and amazing partners. So thank you, take care, be well, and we will be in touch.